Hello students, today I am going to describe the remaining part of life history of Spirogyra. Learning point is sexual reproduction. Asexual reproduction is already discussed in part 1. Sexual reproduction. Sexual reproduction takes place during unfavorable conditions. Unfavorable conditions like low levels of nitrogen in water, high light intensity, and lack of water stimulates the sexual reproduction in Spirogyra. Any cell of the filament can act as a reproductive cell or gametangia to make a gamete. For the fusion process, one tube-like structure is formed during the conjugation process. It is known as conjugation tube. This type of reproduction is known as conjugation. Conjugation may be of two types: scalariform conjugation and lateral conjugation. Scalariform conjugation takes place between two filaments of different physiological strains, whereas lateral conjugation takes place between cells of same filaments. It can be of two types: direct and indirect. In direct conjugation, takes place between two adjacent cells and the pore formation takes place in the septum. Whereas in indirect lateral conjugation, the conjugation tube formed due to the papillae-like outgrowth on either side of the septum. Let us discuss about scalariform conjugation. It is the most common method in most species of Spirogyra. It takes place in night when two filaments come close and lie opposite and parallel to each other. Now, thus opposite cells developed outgrowth called conjugation processes. At the point of contact, the tips of this process is dissolved by the act of catalase and cellulase. After that dissolution, it forms a conjugation tube-like structure between two opposite cells. This whole structure resembles as a ladder. Hence, the conjugation is called scalariform conjugation. It is Greek word scala means ladder, forma means shape or egg-shaped conjugation. So here you can see the figure of it. Okay. So here in the first figure, two filaments come side by side, and then a small outgrowth is developed from the wall of these two filaments. Okay, after that, conjugation tube is or conjugation canal is developed by the dissolution of the that papill-like structure. Then this male gamete fused with the female gamete. Now, after the process, one filament is totally blank and one filament is full of this zygospore. Okay. Then let us discuss about lateral conjugation. It is of rare occurrence and it involves the fusion of gametes from two adjacent cells of same filament. Okay, it is of two types: indirect and direct lateral conjugation. So let us see one by one. So in indirect lateral conjugation, during this process, this papillae-like structure is formed in the transverse septum. After this process, the male gamete fuses with the female gamete through the pore in the papillae. Okay. During this process or after this process, the male gamete fuses with the female gamete to form a zygote, and these zygotes form in alternate cells. Like one cell is empty and the another cell is full of zygote. Then one cell is empty and the another cell is full of zygote. So this is known as indirect lateral conjugation. This type of lateral conjugation is also called chain conjugation. Direct lateral conjugation. Let us see what is happening in that. In the Spirogyra jogensis, such conjugation takes place in between two adjacent cells of the filament, in which the upper cell, function as male gametangium and the lower cell as female gametangium. During the process, the lower cell increases in size, but the upper cell remains smaller. The protoplast of the male cell forms an outgrowth which elongates and come in contact with the transverse wall which is present in between these two cells. Later, this male gamete passes through this transverse wall through a small opening. Thus, without formation of conjugation tube, the contact of male cell passes into the female cell, and the fusion of this male and female gamete form a zygospore. Let us discuss about zygote or zygospore. Structure of zygote or zygospore. Zygospore is the only diploid phase of the sexual life cycle. The decay of female gametangia causes the liberation of zygospore. After that, it remains dormant in the pond of the bottom till the favorable condition return. Zygospore wall is thick and differentiated into three layers. Outer layer is exosporium, which is made up of cellulose. Middle layer is mesosporium, which is made up of cellulose and chitin. And inner layer is endosporium, which is made up of cellulose. Here you can see the structure of zygospore. Germination of zygospore. On the return of favorable condition, the diploid nucleus of zygospore undergo meiosis to produce 4 haploid nuclei. Out of these 4 haploid nuclei, 3 nuclei are degenerated. Only one surviving haploid nucleus is there. The zygospore with one haploid nucleus gradually enlarges and bursts open to release a germ tube. The repeated transverse division of germ tube which form a new young filament. Alternation of generation in the life cycle of Spirogyra. In the life cycle of Spirogyra, alternation of generation is haploid generation and diploid generation.
The haploid generation or the haploid phase is long, but the diploid phase is very short lived. The diploid phase is restricted within the zygospore only. Here you can see the diagrammatic life cycle of Spirogyra. Thank you.